Massively parallel characterization of transcriptional and regulatory elements in 3 diverse human cell types.
Okay, thank you all for attending and for inviting me here. Today I'll be presenting some research that I began about 5 years ago, back when I was a postdoc in Jay Shinduri's laboratory. And this work reflects a collaborative effort between the Shinduri lab with the Nadav Ayatov's lab and Martin Kircher's laboratories. Just to begin with a little bit of background information, of course, during the many years of ENCODE that have undergone so far, we've seen the diversity of different biochemical assays that have shown us that the existence of possibly over 1 million different regulatory elements sprinkled across the genome. And this leads to a pretty daunting challenge because it's led to a little bit of a controversy of whether these elements are actually functional in some way. Someone who's a little bit more liberal in their assessment might think that if you see a biochemical mark, it might reflect something that's functional. And someone that's more conservative might say that, well, you really need to prove that there needs to be some sort of phenotype associated with this to be functional. So along that continuum of liberal to conservative, we're kind of trying to think of alternative assays that can maybe inform us something about the function of these putative regulatory elements that have been defined. And so our perspective into the problem was to use this classic episomal reporter vector, in which you can snip out a piece of the genome and then clone it in a reporter vector, and then evaluate its impact on the transcription of the reporter. And the nice thing about this type of layout is that you can parallelize it into a massively parallel format so that we can potentially test thousands of different putative regulatory elements at once. So in this project, we wanted to address several overarching questions. First question was, can we improve lenti-MPRA technology to robustly measure most of the genome's promoters and putative enhancers in multiple different cell types? This has historically been somewhat challenging because of the scale of lenti-MPRA in the past has been a little bit limited to about approximately 5,000 to 20,000 different elements in parallel. And so we're trying to scale this up about fourfold more than what had been possible in the past. And secondly, we'd like to ask, with this data in hand, can we develop quantitative models to better predict an element's regulatory activity, as well as its cell type specific activity? So we developed over the years this lenti-MPRA technology, which is a flavor of MPRA that allows the readout of the element's activity in the genomic context, because due to the integration of these elements into the human genome. And this is in contrast to many other flavors of MPRA that rely on episomal type of readouts. And so the general strategy of how this assay works is that you can order a group of oligonucleotides, approximately 200 nucleotides in length. And with the first round of PCR, you can then add a minimal promoter. With the second round of PCR, you can then add in random barcodes associated to these elements. And you can clone this into the PLSMP vector to arrive at your element library. Now, the first step here is that we really need to understand which elements are associated with which barcodes, because the barcode will be transcribed in the 5' UTR, in this case, of the gene itself. And so we can actually sequence this library to reconstruct kind of like a dictionary or a hash table mapping which barcode is associated with which element. And subsequent to this, we can use our library to package it into a lentivirus, transduce it into cells, and perform this experiment in triplicate, such that we can measure and sequence the number of barcodes, both at the DNA level and RNA level. And by sequencing this and then quantifying the counts of at the DNA and RNA level, we can then look at the ratio between the RNA and DNA and take the log base 2 transformation of that as a kind of like an element activity score. And so that tells you per molecule of DNA in the input, how many molecules of RNA were expressed as a consequence. So I just wanted to give you a feel for how this data looks like with one of the first pilot libraries that I designed of only about 7,500 elements. So in this particular library, I designed it to have about 6,400 different putative enhancers defined as regions of DNA successibility or open chromatin in the genome. And I coupled this with different types of controls. So some of these were negative and positive controls isolated by Gasparini et al. Or Folco et al., which are two CRISPR inhibition type screens. I also used dinucleotide shuffled controls as a negative control. And then we used some of the controls from Jason's study, who's the co-presenter here, who inspired some of the controls to validate whether the ones that he discovered in the past in his MPRA could be validated in our MPRA. And so just to give you a feel for this, in the beta globin locus, I selected 5 different enhancers in this region. This is one of my favorite loci in the genome because it's been well studied for 30 years or so. And so I selected each one of these enhancers called HS1, 2, 3, 4, 5, as shown in the first row here. And for each of these enhancers, we can then quantify its level of effect on the reporter expression. And we can see clearly that HS2 has the highest activity of them all, whereas HS5 has the lowest activity. So it kind of gives us a quantitative feel for, in isolation, how much these elements can activate transcription. And I want to mention that HS2 has been in the past, through genetic experiments, through CRISPR inhibition experiments, et cetera, over and over been isolated as the #1 candidate hit in this locus as the top active enhancer of this locus control region. So our data were consistent with what had been seen previously. And so now we wanted to try something a little bit more ambitious. We wanted to see, can we expand the scale of this lenti-MPRA much farther than we could before? So in particular, we wanted to start designing experiments where we test all the DNase peaks in the genome that are intergenic. And so they overlap, that's kind of our definition of putative enhancers here. In addition, we'd like to also test all the promoters in the genome associated with protein coding genes, centered at the transcriptional start site of that gene. So we can test each of these different regions in both orientations. For enhancers, we can test them the forward and reverse orientation relative to the reporter. And for the promoter, we can test it in the sense and antisense orientation relative to the reporter. And by pooling all these together and testing them as 200 nucleotide elements, we can then perform the typical sequencing and then quantify the element activity score by the RNA to DNA ratio here. So how did I design these initial experiments? For HepG2, for example, we were able to test approximately 164,000 elements. And in particular, in this design, we had forward and reverse orientations of about 66,000 DNase peaks corresponding to our putative enhancer set. In addition, we tested 19,104 protein coding gene promoters in the sense orientation, and the majority of that in the antisense orientation as well, the 12,411 in the antisense orientation. We also tested dinucleotide shuffled negative controls, as well as synthetic controls engineered in a previous experiment. Now, in addition to this, I also designed libraries for K562 cells as well as WTC11 cells. For K562 cells, we designed approximately 87,600 enhancers in both orientations. We tested all protein coding genes promoters in both orientations again. We also looked at 7 different loci that had disease biology relevance, and the ±1 kb locus around those regions. We tested different 200 nucleotide fragments in the intergenic space there to test if whether regions outside of DNase hypersensitivity sites could potentially also have activating or repressive effect. In addition, we used dinucleotide shuffle controls, and again, controls chosen by Jason Ernst's prior study. And now, and finally, in WTC11 cells, this cell type is a little bit more tricky because it has a more limited transduction efficiency. So we were able to only test about 75,000 elements. And in particular, the breakdown of this library was about 30,000 putative enhancers tested in both orientations, as well as 7,500 promoters tested in both orientations. And so how did this data look overall? So here I'm plotting the distributions of the MPRA data now on the y-axis. And we can see the distributions relative to the dinucleotide shuffled random controls. And sure enough, our negative controls from the Smith et al. Study show a negative signal similar to the shuffled controls, whereas those that are positive controls, we can see very clearly a much stronger signal for transcriptional activation relative to our shuffled controls. promoters and putative enhancers are kind of somewhere in between. For promoters, we can see a bimodal distribution emerge, whereas for putative enhancers, we can see this right-skewed distribution emerge. And we saw something similar no matter which cell type we looked at. Now, I mentioned earlier that there's a little bit of a controversy about how many enhancers are functional. And so this approach actually gives us a quantitative measurement to actually get at that question, at least from the perspective of transcriptional gene regulation in these cell types. So what we can do here is we can quantify how many of these elements basically exceed the 95th percentile of the shuffled set of sequences. And that gives us a 5% false discovery rate, but it tells us approximately how many enhancers and promoters are quote unquote functional. And that is beyond the level of expression conferred by random sequences. And so we can see that for promoters, approximately 55% of sequences were active in HepG2 cells, 52% approximately in K5X2 cells, and 51% approximately in WTC11 cells. Whereas for enhancers, we discovered that approximately 43% of them were functional in HepG2 cells, similar percentage in K5X2 cells, and a lower percentage of approximately 26% in WTC11 cells. So next, what I wanted to evaluate is other properties of the promoters that we were evaluating. So I mentioned that we chopped out these 200 nucleotide fragments of promoters and tested them in both orientations. And so there's more additional data presented in the paper, but for now, I'll just present the core discovery, which is that if you just look at the expression conferred of these small core promoters in the massively parallel reporter assay shown on the x-axis, they actually correlate decently strongly with endogenous gene expression as shown on the y-axis. So on the y-axis, we can measure that by the transcripts per million, as measured from an RNA-seq experiment, for example. And we can see that there's a correlation here of approximately 0.57, which means that this result was a little bit surprising to me because it's been a little bit debated in the literature how long a core promoter might be. And this is kind of showing that even a core promoter as short as 200 nucleotides still possesses a tremendous amount of information in specifying gene expression levels. Next, I'd like to turn to a slightly different topic, the question of the orientation dependence of regulatory elements. So as I mentioned, we tested all these different elements in multiple different orientations. And so we can then quantitatively assess whether there's a signal here that tells us that there's an orientation dependence. And so my way of approaching that question was to look at from how different replicates behaved. So if we look at the forward orientation of replicate 1, for example, relative to the same orientation tested in a different replicate, replicate 2, and do that comparison all three different pairs of replicates for both the forward versus forward orientation or reverse versus reverse orientation, that is, the orientation is the same, we can see a Pearson correlation of about 0.95 or so. But now if we perform this comparison looking at the correlation of replicates in the opposite orientation, that is the forward orientation of the first replicate versus the reverse orientation of the second replicate, or vice versa, we can see a much lower correlation than we've seen when the orientation is matched. And this demonstrates now that there is actually some sort of orientation dependence, and we saw this phenomenon in all the different cell types that we examined. So a second way of looking at this is if we average across all the different replicates that we have and look at the forward orientation on the x-axis now and the reverse orientation on the y-axis, we can see by and large there's a pretty strong correlation here, a correlation of about 0.74. But nevertheless, there are these off-diagonal elements here that seem to have greater activation ability in one orientation relative to the other orientation. So this shows that although there's not too much of an orientation-specific effect, we can still detect quantitatively that there exists an orientation-specific effect. Next one I'd like to turn to is some benchmarking to try to predict the type of data that we've measured. So my approach to this was to test several different types of regression models. The first was a biochemical regression model trained using lasso regression. And the input features to this model were a group of DNA-seq types of activity scores in these regions, as well as histone ChIP-seq of different histone marks such as H3K27 acetylation or trimethylation, et cetera, as coupled to ChIP-seq of different transcription factors. And when we put all these together, matched to a particular cell type, you can train a model together to see which biochemical marks are most predictive of the MPRA data. In conjunction with this type of approach, we also were interested in sequence-based approaches. That is, if the model knows nothing more than the input sequence itself, what can the model learn just from the sequence in predicting the element activity? So our approach here was to use a deep convolutional neural network called MPRA-NN, consisting of 4 convolutional layers. It's a pretty simple neural network overall, and its input is just a one-hot encoding of the sequence itself. And here it's being trained to predict the MPRA activity. The third title model that I wanted to test or benchmark was a model that I'd previously worked on during my time at Calico. Here we trained a model called Informer, which is trained on basically the entire genome, and it outputs predictions of over 5,000 different biochemical tracks. So it's trained on a much larger data set, and we'd like to see if we can use what it's learned to fine-tune it on this new type of task of predicting MPRA activity. So in showing this benchmark, we can see the biochemical model in red. It turned out that the sequence-based model trained from the convolutional neural network in purple was able to outperform on these 10 different held out folds of data relative to the biochemical model. And we also saw that the Informer model seemed to outperform the MPRA-NN model. And so this pattern seemed to hold true for all 3 different cell types, in which we saw kind of a range of Pearson correlations from about 0.73 or so to about 0.86 or so. And so although the Informer model seems to be doing better, there are a large number of disadvantages of this model. And so one of the disadvantages is that it was pre-trained on the entire genome, so it's a little bit tricky. Even though it has never seen MPRA data in particular, it's still seeing some of the elements that we're assessing in our test set. And so you could say it might be a little bit inflated in its performance. Moreover, it's a little bit disadvantageous because it consists of over 5,000 different features. And this feature set has tremendous multicollinearity, making it difficult to distinguish which of these features are most relevant. Mechanistic interpretation is very difficult as well with respect to transcription factors. So for that purpose, I was a little bit more keen on modeling the MPRA neural network, or MPRA-NN, which is a little bit easier to tackle and interrogate from the perspective of transcription factor binding sites. And so luckily, Anshul Kundade's lab has developed a really nice tool developed by Jacob Schreiber called TF Modisco Lite, and we utilized that tool to interrogate what the neural network has learned. And so by looking at the sort of predictions and tracing back the binding sites that are most predictive of the data, we could basically reconstruct 3 major global binding sites that seem to be relevant in all 3 data sets. So KLF 515, which is a known stripe factor that potentially recruits other transcription factors, FOX I1 and J2, and NFY, C, and A seem to be the top 3 factors universal to all 3 different cell types. But in addition, we found certain factors that we never saw again in other cell types. So for example, in HepG2, we saw the HNF4A and 4G motif emerge. For K562, we observed the GATA2 and 3 motif emerge. And for WTC11, our number 1 motif was the POU5F1 SOX2 motif. And all 3 of these motifs are well understood as lineage-specific transcription factors. So it was really reassuring to see that our neural network could automatically learn these motifs without having any knowledge of the motifs at first. I'm just showing you a few motifs here, but in the supplement of the paper that we've deposited on bioRxiv, there's a large number of other interesting motifs that seem to have emerged as well. The other nice thing about the MPRA-NN model is that it's a much more compact model. It's easier to use the model to generate predictions on genetic variants, for example. And so that's what kind of the next task that I was really interested to turn to, just to evaluate what we can learn from MPRA data. Can we extend into predictions of genetic variants and their effects. So there's this really nice study by Martin Kircher a few years ago in Nadav's, working with the Nadav, in which he performed a saturation mutagenesis and coupled to 20 different disease loci, and coupled this to a massively parallel reporter assay to actually measure for each nucleotide variant, what is the effect of that variant on transcription of the reporter? And so we have data now for every single base mutation here of different promoters and enhancers. In this particular case, I'm showing you the sort1 enhancer in particular. And so a little green arrow here indicates a genetic variant that was significantly, led to a significant change relative to baseline. And so shown on the top here is my personal annotation of this data. We could see several motifs emerge that I was able to annotate using TomTom. And next, we can just look at how does MPRA-ANN predict this type of data. And certainly the predictions aren't perfect, but there's at least a strong positive correlation of 0.47 or 0.5 Spearman correlation here. So it's able to detect some of these motifs pretty well. So you can see the first 5 here are detected pretty well. Some of them are, however, missed. So you can see it does have a false negative rate. So it attributes these 3 or 4 different transcription factor binding sites as not having much of an impact. But in addition, it has a greater prediction in effect size for 2 motifs downstream that don't have too much of an impact, but they're definitely statistically significant. So it's telling us real motifs that seem to have relevance, but maybe exaggerated their effect size relative to what the saturation mutagenesis tells us. So I've shown you data for 1 locus here, but there are 2 additional ones I've presented in the supplementary data that you can check out as well. Finally, I'd like to turn to our last experiment here, which was interrogating whether we can actually test the same cohort of elements in multiple different cell types to start getting the question of cell type specific element activity. And so we designed a joint library of 60,000 elements consisting of 19,000 putative elements, enhancers in HepG2 cells, 19,000 putative enhancers from KFX2, and approximately the same number in WTC11. These are derived from DNA peaks in those respective cell types. A common set of about 2,400 promoters, 300 dinucleotide shuffle controls, and some other controls here. And so, as I mentioned before, we've now used this exact identical library, transduced it in 3 different cell types, and measured the impact using MPRA. And that tells us, allows us to measure the cell type specific activity. And so, because I don't have much time here, I'll just show one major result here, which is if we remove the first principal component and look at the cell type specific components of regulatory activity measured by the MPRA, we can plot it as this biplot, showing the loadings in the middle here, along with PC2 and PC3. So we can see that the cohort of elements that were positive controls in HepG2, sure enough, they were very specifically active along this HepG2 loading, whereas the positive enhancers derived from the Ernst et al. Study were very specifically active in K562 cells relative to the other cell types. There are also a large number of putative enhancers that were pushed a little bit in that direction, but with perhaps less cell type specificity than we had maybe expected. Nevertheless, we were able to train a machine learning model, again, a neural network model, to predict the cell type specific behavior of these elements, and interpret that model to interpret or dissect which regulatory motifs might explain the differences in these different cell types. So for example, in HepG2, we discovered the top 3 motifs for HNF4A or 4G, BATF3 or BAC1, and HNF1B and A. In contrast, in KFX2 cells, we discovered the GATO1 TAL1 motif, STAT145A, EHF, and ELF3. And curiously, in WTC11, we discovered CTCF as one of the top motifs showing cell type specificity, SOX2 POUF 5F1, as well as SOX3, 4, and 6. So these are our top 3 motifs, but there are additional repertoire of cell type specific motifs we discovered in addition to these. So just to wrap up here, I've shown you that we've further improved the scalability of lenti-MPRA technology, enabling us to test approximately on the order of 680,000 transcriptional regulatory elements collectively among 3 cell types. And this encompasses about 19,000 protein coding gene promoters and 183,000 candidate enhancers tested in both orientations. We were able to observe approximately 40% of enhancers functionally activate transcription. We detect a subtle but detectable signal of element asymmetry, and find that the human 200 nucleotide core promoter strongly explains endogenous gene expression among protein coding genes. Moreover, we were able to see that sequence-based models seem to outperform biochemical models in predicting MPRA data. And we're able to interpret these models to define specific transcription factor binding sites, which we believe modulate gene expression also in a cell type specific way. And all of the data for this work has been processed. I've deposited along with the preprint, and you can discover it with the bioRxiv article if you'd like to analyze the data yourself, and the MPRA NN code is freely available as well. Finally, I'd just like to acknowledge all the people involved, including the Kircher groups, Jay Shinduri's group, Nadav's group, Taka, who generated all the experimental data behind this, and Bill Noble and Gurkhan Yuradimji. Thank you very much, and I'd be happy to take any questions.